Welcome to today's lecture on the topic of apoptosis and programmed cell death. Apoptosis is a highly regulated process of cell death that plays a crucial role in various physiological and pathological contexts. In this lecture, we will explore the molecular mechanisms underlying apoptosis, its significance in development and tissue homeostasis, its implications in various diseases, and the pharmacological strategies that can be used to target this process. Furthermore, we will discuss the techniques used to study apoptosis, the crosstalk between apoptosis and other cellular processes, the epigenetic regulation of apoptosis, and its interactions with the immune system. Moreover, we will delve into the computational modeling of apoptosis and its applications in regenerative medicine. Finally, we will touch upon the ethical considerations and the public perceptions surrounding apoptosis. Now, let us venture into the intricate world of apoptosis, starting with the basics. Apoptosis, or programmed cell death, is a carefully orchestrated process that allows cells to self-destruct in a controlled manner. Unlike necrosis, which is an accidental and unregulated form of cell death, apoptosis serves essential physiological functions, including embryo development, tissue remodeling, and the maintenance of tissue homeostasis. It also acts as a defense mechanism against the proliferation of damaged or potentially harmful cells. At the molecular level, apoptosis involves a complex interplay of signaling pathways, protein interactions, and gene regulation. Key players in this process include caspases, a family of proteases responsible for initiating and executing various steps of apoptosis. Additionally, the BCL2 family of proteins regulates the balance between pro-apoptotic and anti-apoptotic signals, thereby determining cell fate. The molecular mechanisms of apoptosis can be divided into two major pathways, the intrinsic pathway and the extrinsic pathway. The intrinsic pathway is activated by intracellular stress signals, such as DNA damage or oxidative stress, and is regulated by the mitochondria. The release of cytochrome C from the mitochondria triggers the assembly of the apoptosome, a multi-protein complex that activates caspase cascades, leading to cell death. On the other hand, the extrinsic pathway is initiated by extracellular signals, such as cytokines or death ligands, binding to specific cell surface receptors. This triggers the assembly of the death-inducing signaling complex, DISC, which activates caspases, ultimately leading to apoptosis. Both pathways converge at the execution phase, during which caspases cleave various cellular substrates, resulting in characteristic apoptotic morphological changes, such as cell shrinkage, nuclear condensation, and fragmentation. Apoptosis plays a fundamental role in embryonic development, ensuring the proper formation of organs and tissues. For example, apoptosis is critical during the shaping of body structures, the removal of excess cells, and the elimination of interdigital webs. Furthermore, apoptosis is essential in the maintenance of tissue homeostasis throughout life. It regulates the turnover of cells in continuously renewing tissues, such as the skin, gastrointestinal tract, and hematopoietic system. By eliminating aged or damaged cells, apoptosis contributes to tissue integrity and prevents the accumulation of aberrant cells, which could lead to tumor genesis. Dysregulation of apoptosis is implicated in various diseases, including cancer, neurodegenerative disorders, autoimmune conditions, and cardiovascular diseases. In cancer, evading apoptosis is a hallmark feature that allows tumor cells to evade immune surveillance and acquire resistance to therapy. Neurodegenerative diseases, such as Alzheimer's disease and Parkinson's disease, are characterized by the accumulation of misfolded proteins and the failure of cells to undergo proper apoptosis, leading to the progressive loss of neurons. Autoimmune conditions, such as lupus and rheumatoid arthritis, involve an imbalance between pro-apoptotic and anti-apoptotic signals, resulting in the destruction of healthy tissues by the immune system. In cardiovascular diseases, apoptosis contributes to the death of cardiac cells during ischemia, reperfusion injury, and heart failure. Understanding the molecular mechanisms underlying apoptosis in these diseases opens avenues for therapeutic interventions. Given the significance of apoptosis in various diseases, pharmacological targeting of this process has emerged as a promising therapeutic strategy. Numerous drugs have been developed to modulate apoptotic signaling pathways, either by activating pro-apoptotic factors or inhibiting anti-apoptotic factors. For instance, small molecule inhibitors of anti-apoptotic BCL2 family proteins, such as venetoclax, have shown efficacy in treating certain types of cancer. Additionally, activation of death receptors or stimulation of the intrinsic apoptotic pathway can be achieved through the use of agonistic antibodies or pro-apoptotic peptides. However, the therapeutic targeting of apoptosis is complex and requires careful consideration, as excessive induction of cell death can have detrimental effects on normal tissues. Therefore, a thorough understanding of the specific context and mechanisms of apoptosis in different diseases is crucial. Studying apoptosis necessitates the use of various experimental techniques to unravel its intricate mechanisms. Several methods are available to detect and quantify apoptotic cells, including microscopy-based techniques, such as DNA fragmentation assays and Annex V staining, which allow the visualization of apoptotic morphological changes. To investigate the molecular players involved in apoptosis, techniques like Western blotting, immunofluorescence, and flow cytometry are employed to detect and quantify apoptotic proteins and their post-translational modifications. In recent years, advanced techniques, such as single-cell RNA sequencing and proteomics, have provided valuable insights into cell-to-cell heterogeneity and the regulation of apoptosis at a systems level. Apoptosis does not occur in isolation but is intricately linked to various other cellular processes. One prominent example is the interplay between apoptosis and inflammation. The release of intracellular components during apoptosis can trigger an immune response, leading to inflammation. Conversely, inflammation can modulate apoptosis, either promoting or inhibiting cell death, depending on the context. Furthermore, apoptosis can interact with processes such as autophagy, senescence, and DNA damage repair. These interconnected pathways influence each other's outcomes and collectively shape cellular fate decisions. Epigenetic modifications, including DNA methylation, histone modifications, and non-coding RNA molecules, have emerged as critical regulators of apoptosis. They can influence the expression of pro-apoptotic and anti-apoptotic genes, thereby modulating cell death pathways. For instance, aberrant DNA methylation patterns have been associated with the dysregulation of apoptosis in cancer. Histone modifications, such as acetylation and methylation, also play a role in regulating gene expression during apoptosis. Moreover, non-coding RNAs, including microRNAs and long non-coding RNAs, can act as both promoters and inhibitors of apoptosis by targeting specific mRNA transcripts. Understanding the epigenetic regulation of apoptosis provides insights into disease pathogenesis and potentially opens new avenues for therapeutic interventions. Apoptosis and the immune system are intricately connected, with cell death regulating immune responses and immune cells influencing apoptosis. Apoptotic cells can be engulfed and cleared by phagocytic cells, preventing the release of potentially harmful intracellular contents and dampening inflammation. Furthermore, apoptotic cells act as a source of antigenic material, promoting immune tolerance and educating immune cells. Dysregulation of apoptosis can disrupt the immune balance, contributing to autoimmune diseases or impairing immune responses against infections or cancer cells. Computational modeling has become an invaluable tool for studying complex biological processes, including apoptosis. Mathematical models can integrate experimental data, simulate dynamic behaviors, and provide insights into the underlying mechanisms of apoptosis. By representing the interactions between key molecular players mathematically, computational models allow us to explore the effects of perturbations, predict the responses to different stimuli, and identify potential targets for therapeutic intervention. Moreover, they can contribute to our understanding of the emergent properties of apoptosis, such as cell population dynamics and tissue-level behavior. The manipulation of apoptosis holds great promise in regenerative medicine. In tissue engineering and regenerative approaches, controlled induction of apoptosis can facilitate the removal of damaged or diseased cells, making space for the integration of regenerative cells or biomaterials. Additionally, modulating apoptosis can influence the fate of stem cells during differentiation, allowing precise control over tissue development and regeneration. Understanding the interplay between apoptosis and regenerative processes is vital for harnessing the potential of apoptosis in tissue repair and organ transplantation. As with any scientific advancement, the ethical considerations and public perceptions surrounding apoptosis warrant attention. While apoptosis is a natural and essential process, the manipulation of cell death raises ethical questions. In some cases, the intentional induction of apoptosis may raise concerns about its potential misuse as a tool for selective elimination or alteration of specific cell populations. Additionally, public understanding of apoptosis and its applications may impact societal acceptance and adoption of related technologies. Therefore, as researchers and practitioners, it is crucial to engage in ethical discussions, ensure transparency, and educate the public on the benefits, limitations, and potential risks associated with the manipulation of apoptosis. In conclusion, apoptosis is a fascinating and complex process that plays a critical role in various aspects of biology and medicine. From its involvement in development and tissue homeostasis to its dysregulation in diseases, understanding the molecular mechanisms underlying apoptosis opens avenues for therapeutic interventions. The exploration of techniques, such as computational modeling, provides a deeper understanding of the complexity and interconnectedness of apoptosis with other cellular processes. Furthermore, the ethical considerations and public perceptions surrounding apoptosis underscore the importance of responsible scientific endeavors and effective communication with the society at large. As we conclude this lecture, let us reflect on the remarkable intricacy and significance of apoptosis, and how its understanding continues to shape our understanding of life and disease.